Welcome to our presentation on computational analysis on electrocardiogram readings. This is by Fungi, and we're joined by Lockery and Ryan, Andrew, Che, and Kaslin. So first off, what is an electrocardiogram? These are most commonly known as ECGs and EKGs. It's an electrocardiogram records heart signals. It can be used to identify heart issues quickly and monitor the heart. And it measures electrical activity, notably the depolarization and repolarization, which contracts heart muscles. It's measured by an array of electrodes put onto the body surface. The different waves represent the depolarization and repolarization of the atria and ventricles.
So why should we be using computational approaches as opposed to like a physician just kind of reading it for you? This allows for better patient care. Cardiologists don't have to, be with every patient if there's a machine kind of doing its work for them. But this only works when the computer's interpretation is very close or at what a trained expert would do. And that turned out to be pretty true. 98.4% of the results are exactly the same as the cardiologists. And, a very small percentage right there, as you can see, would result in a different recommended care action.
And so next up is a computational pipeline for analyzing an ECG. So first off, there's feature extraction, which detects certain features of the analysis, such as QRS and other special features. And then after that is feature selection, which removes some of the redundant data and only keeps the important data for better computational speed and efficiency. And then finally, it's classification of the data to provide better results and analysis.
All right, so the very first step before we even classify and extract the features is the denoising, or detecting the actual ECG signal. This is far from a simple task because, for example, breathing and that'll interfere with lower frequency parts of the ECG, and like your other muscle activity besides your heart will interfere with the high end. So we start with sort of the higher graph on the right with the picture there, and we need to get to the lower one. And there's a few different methods of doing that. A hard threshold, which is just sort of cutting out anything that like oscillates too much. That ends up having a noisier end graph, but it's a little bit more accurate. The soft threshold, which will adjust its threshold as the ECG continues, produces a smoother graph, but is a little bit less accurate. And then a sigmoid function-based threshold, which sort of combines the two, adds a little bit of algorithmic math, and that results with a lot better performance than either of those two.
Next up, the second step of the pipeline is feature extraction. So the goal of this step is to extract as much information as we can from the data. And for instance, there's a lot of information that you can extract from just a pulse. So one of the common features to extract is the P, QRS, and T waves, which represent the stages of heart contraction. So for the P wave, which is the beginning pulse, is the depolarization of the atriums, which signals the contraction of the atriums. Then the QRS complex is a rapid series of 3 different waves that come right after each other. And they represent the depolarization of ventricles, which also comes right before contraction. And then the T wave is ventricular repolarization, which happens after contraction. And then a Fourier transform is applied to these feature vectors to change the vectors from the time domain to the frequency domain. This is used to reduce computational time and find feature points. Next, the second part of extracting data is dimensionality reduction. So generally, we want our data to have a pretty well approximation of the heartbeat. But at the cost of this, it comes at the cost of having a high computational time. So what we want to do is to perform a trade-off between the approximation of the input signal and the efficiency of our pipeline. And so that's what many algorithms such as PCA do. So what PCA does is that it tends to map original features into a lower dimensional space and gets rid of redundant data, which makes processing the data much more efficient. So there's many steps in PCA. So the first step is standardizing variables so that each of them contribute to the signal equally. The second one is a covariance matrix, which compares the variables, compares the variables to see how correlated they are. Because if it's really correlated, then it might cause redundant information, and that might cloud up the processing time. The covariance matrix is shown on the bottom right. Next, we want to get the eigenvectors and eigenvalues because we want to plot into a new dimensions. So basically, an eigenvector is a vector that changes in magnitude but not in direction after it gets changed into a new dimension. And an eigenvalue is the coefficient of the vector by how much it changes in magnitude. So for instance, in the equation ax equals lambda x, x represents the eigenvector that does not change in direction, and it represents our data of our features because we don't want it to change once we change the dimensions. The lambda represents the eigenvalue, which represents how much it changes in magnitude, so that we store our information instead of having it altered. And then the A is a matrix that represents the shift in different dimensions. And then after that, we use our feature vector, which is a matrix of eigenvectors and our accepted components. And finally, we plot our data into a new space and dimension.
All right, so the final step of the pipeline is machine learning classification. This can be done by a cardiologist, but the point of having a machine do it is so it doesn't have to be one. There's lots of methods to do this. For example, the sort of most simple, the k nearest neighbor groups data points together. It works well if there's not that many data points, but as soon as it becomes more complicated, the resources needed increase quite exponentially. The decision tree is very easy to read and very easy for a different part of the computation to understand, but it only works well with normal heartbeats, which is kind of a problem if you want to be detecting abnormal heartbeats. Support vector machines are useful, they're fast, but they only usually work with smaller samples of data, so they can't be applied as widespreadly. The best method is an artificial neural network, and a deep neural network, which has more hidden nodes between each shown decision, are even better at doing this. But the problem with them is they don't learn all they need to know on their own. The data scientists kind of need to train them a little bit more than we would like for a machine learning algorithm to do so. And the only data that's passed to them is data that scientists know that these neural networks can deal with pretty well, which is not really optimal.
So unfortunately, electrocardiograms are not foolproof. There are quite a few limitations. In fact, from one study, it states that computer ECG interpretation related to life-threatening conditions, like high-degree AV blocks, is frequently not accurate, 75% in this case. So one of the most common technical problems is the arm bead reversal. So as you can see in the picture, when the top line, it looks kind of reversed in the second part. So that's what happens when the two patches are reversed on the arms. And one of the most frequent errors in computer interpretations of ECGs are arrhythmias and pacemakers, which is unfortunate because arrhythmias are dull, they are irregular heartbeats and things like that. So the common misdiagnoses come from electrodiagram artifacts. ECG artifacts are electrodiagram alterations not due to cardiac or electrical activity. Baseline and waves can be distorted as a result. ECGs do use filtering to eliminate unwanted artifacts and or noise, but they do tend to slip in sometimes. So errors in ECG readings can be due to ECG setup, machine settings, or outside interference.
And these are our sources. Thank you for listening to our presentation.